Yeah, I think this paper is basically on the main topics that of a lot of research in molecular oncology, the integration of biomarkers for helping patient diagnosis and therapy. Basically, what this paper is saying, it's saying that a protein coding gene marker can be integrated with microRNAs, which are short non-coding RNA markers, in order to identify how the patient with breast cancer are responding to the treatment. This is on a larger topic that it's very hot now in molecular oncology, topic of having cancer as a disease of coding genes and non-coding RNAs. Most well-studied are microRNAs, and basically microRNAs are one of the main topic of the conference that it's organized these days in Basel. Yeah, basically, the concept is non-coding RNA field. It's very hot. There are over 10,000 papers on microRNAs each year and over 2,000 papers already on long non-coding RNA. The concept of this journal is to try to attract very interesting papers in the field to have a good impact factor and a lot of citation. Of course, it's a big challenge in the new type of publishing where a lot of journals who are offering space for publication, but I think with a very distinguished set of editorial board and with a lot of good publications in the next few years, the journal will survive to the competition and will be on the market in publishing good work on non-coding RNAs. Everybody who thinks has a good paper on this topic is welcome to send to the journal, having in mind that we are using a very strict review process, so the papers are accepted or rejected according to scientific merit.
The first clear-cut translation come from our studies of MIR15 and 16. You remember, in 2002, George Calin and I found that in CLL, the most common and dominant genetic alteration is the loss of two microRNA, MIR15A and MIR16-1. And that was the first evidence that the non-coding genome can contribute to disease, in particular to cancer. Before 2002, when we published this paper, everybody was convinced that only oncogene and tumor suppressor gene were involved in cancer pathogenesis. In other words, genes that encode for protein. Our paper was the first evidence that alteration in the non-coding genome can result in disease, in particular in cancer. That was a revolutionary discovery because it indicates that 90% of the genome that was considered garbage is not garbage. Not only is transcribed, but contribute to normal physiology and pathology. After that, we looked for target of MIR15 and 16. And we found that the crucial or critical target of this two microRNA is a gene that my lab discovered a long time ago, in 1984, and I call it BCL2. And therefore, it became clear from our result that loss of MIR15 and 16, due predominantly to the lesion, leads to overexpression of BCL2. But at the same time that this was going on, a company in the United States called Abbott developed small molecule inhibitors of BCL2. And after working on the molecule, it took a long time, they developed finally a molecule called venetoclax, or ABT-199. Now, this drug is dynamite. This drug can cause complete remission in patients with CLL, and the drugs kill leukemic cells so fast that there is a crisis because most of the leukemic cells are die. And therefore, we can start using a little concentration of the drug and escalating it. And what is amazing of this drug, that in many of these cases, there is no sign of minimal residual disease after treatment. Now, it took a long time. This drug was approved just last year, in April of 2016, while we made the discovery in 1984. So it took 32 years to go from the discovery of the gene to the development of the drug that can be used and will save, I am sure, thousands of lives. Okay? So that indicates how the understanding of the non-coding genome can contribute not only to develop a very important biomarker to study the disease, but to develop drugs. And I'm sure, and we have plenty of evidence that we have developed during the last few years of research, that we can go from dysregulation of specific microRNA to the development of new drugs and new therapeutic approaches. Now, microRNA and other non-coding RNA, like long non-coding RNA, can be used as very powerful biomarker. They can be used as powerful biomarker when they are expressed in the cell, in the cancer cell, for example, but also by looking at these biomarkers in the blood. And this is a very important issue, because if we can detect aberrant expression of this biomarker in blood very early during the development of the disease, we might be able to cure the disease and eradicate the cancer when the tumor is not invasive and metastatic yet. Okay? So a lot of progress has been made because of the discovery of the involvement of non-coding RNA in human disease.
This is a young field. We're faced with an enormous challenge of understanding the structure and function of literally tens of thousands, if not more, of regulatory RNAs that appear to be overseeing the trajectories of human development and brain function. So the challenges within that new world are to work out how the structure of these RNAs is affecting their function, and also how mutations or variations in those sequences give us predispositions to complex diseases like neuropsychiatric disorders and neurodegenerative disorders and cancer. I think we are embarked upon a huge endeavour. There are many of these RNAs to be analysed in terms of their biological function. We want to understand the principles and the way they work and the different domains in which they work. And also the thing that excites me most is the understanding the superimposition of plasticity on this system through RNA editing and modification, which I think the evidence points very clearly to saying that this is the fundamental platform for the evolution of cognition and brain function. You know, the 20th century was often considered, the second half, as being the genesis of modern molecular biology. It was the genesis, but it was just the warm-up. This is the century of biology and medicine. And when we understand the non-coding RNA world, we'll understand human biology. Well, the most important aspect to consider in non-coding RNA research is the interplay between experimental data and informatic analysis. I think, and not just in non-coding RNA research, I think in science in general, it is essential that practitioners, and particularly students coming into the field, know how to handle big data sets, are confident to commission big data analyses through genome sequencing and transcriptome sequencing, and also confident to do high-throughput genome editing screens to look at the functions of these RNAs. So I think a new tool set is required. I think a new mindset is required. and I think new energy is required.
So I think the conference is a good mixture of different type of scientists involved in many aspects of RNA. RNA is a molecule between DNA and proteins, but the RNA itself, it does many things, it has many functions. And here there is, I would say, a good mixture of scientists studying in detail the basic mechanism of what is doing RNA, the ribonucleic acid, and what are alterations in disease, for example. In our case, for example, we're studying how epigenetics, how chemical modifications are able to alter the expression of non-coding RNAs in cancer cells. It is known that, for example, that there are several tumor suppressors that they act against cancer. The cancer cells deselect the loss. There is a loss of function of these RNAs, and this promotes the growth of tumor cells. The good news about this is that we can detect this. It can be used like biomarkers of the progression of the disease. And maybe, why not, thinking about therapies that are directed to RNA. RNA itself, or just to erase the chemical marks that silence these RNAs. So it's another tool that can be used in the better management of cancer patients.
My name is Menashe Bar-Eli. I came here from MD Anderson Cancer Center in Houston. It's the biggest hospital to treat cancer patients in the United States. And personally, I work on the progression of melanoma and studying what makes melanoma to metastasize. And for many years, we were concentrating on transcriptional regulation of genes, but recently we realized that there is another layer of controlling gene expression, and this is the microRNA, and this is what this meeting is concentrating about. What I like about this meeting is because they invited scientists from different disciplines to talk about these subjects, from how microRNA regulating genes, their heterogeneity, and mostly how mutations within the microRNA can affecting the target genes and contributing to diseases such as melanoma. For example, we found that in melanoma, the metastatic melanoma cells are lacking an enzyme that can regulate editing of the RNA, and the editing of the microRNA can really affect the target genes, hence contributing to the metastatic phenotype of melanoma. Overall, this is a wonderful meeting. They assembled a fantastic group of scientists and speakers to talk about several aspects of how microRNA regulating diseases.
This field has been developing for a number of years, maybe past 15 or 20 years, and it's been very interesting to see the different stages of the development of these non-coding RNAs in disease, where initially there was a great deal of interest in profiling the different microRNAs and non-coding RNAs, and then that's led into various translational applications, including the use of these RNAs as diagnostic biomarkers as well as therapeutics. I think the biggest challenge right now is to see if we can, in fact, develop these in terms of therapeutics, because that's ultimately what we want to do in cancer and other diseases, to be able to treat the disease with the therapeutic. And there's really two different ways that can be done with the microRNAs. One would be to in the cancer, there's one of two situations. Either the microRNAs are reduced in the tumor compared to normal, or they're increased in the tumor relative to normal. So in the case, and both of them can be causative or contribute to the causation of the cancer. So in the case when the microRNA genes are reduced in the tumor, we would then add those back in the form of something called a microRNA mimic. And in the case if they're overexpressed, we'd want to inhibit them with something called an anti-mir. There's still a lot of challenges in the delivery of this of these therapeutics, and I think one of the big exciting, something that's very exciting on the horizon is the use of these exosomes or extracellular vesicles to deliver the therapeutic oligonucleotide to treat the cancer.
Oh, this is a really great conference about non-coding RNA, which is becoming a very hot topic on many aspects of research and translational application. And I think it's a very good reunion on this topic of many scientists that work on that. And it was very interesting and it was fun. The field in which we are working is on the use of short RNA in therapeutics, cancer therapeutics, which is a new field that is promising to give important results for the therapeutic of cancers. We have produced already, we and many other teams have produced already data that shows that even this small RNA, 20 nucleotides, can improve cancer therapeutics. So I think this is a very important field, and it's an application that when we started, and this field started about 10 to 15 years ago, nobody could even imagine.
It's my first time participating in this conference. It's very exciting. A lot of new things are being discussed. Many things are unpublished. It's a very exciting event. Certainly. I work on two types of short non-coding RNAs, the tRNA fragments and the microRNA isoforms. These are non-coding RNAs that regulate the expression of proteins.
I found the conference very exciting because it just adds even more excitement to the non-coding RNAs and RNA in general in regulation of gene expression. I've been working on different aspects of RNA for nearly 50 years, and last 2-3 decades demonstrated that RNA not only functions as a major template for protein synthesis, but also functions as a regulatory RNA, as a short or very short RNA like microRNAs or intermediate length or long RNAs. So I'm excited to find out that this field is really very dynamic and new discoveries are made almost daily, and it looks like there will be no stop to that because there's so many of these non-coding regulatory RNAs that it will take us probably decades to decipher their function. In a way, we have a problem like dissecting a second human or mammalian genome, because protein coding genome, that was one thing. We understand quite a lot about it, but very little about the function of non-coding RNAs. And seeing the progress also presented at this meeting, which also links non-coding RNAs to disease, in particular to cancer. And this is another reason people are excited about non-coding RNAs, because many of them are demonstrated to be cause or associated with disease, not necessarily with causative demonstration, but another important thing is that one is talking more and more about therapeutic use of these non-coding RNAs or their analogs in different human diseases. And I'm sure, of course, it will take another decade to demonstrate utility of these therapeutic approaches, but I'm quite confident that they will eventually show up. Yeah.
It's really a great pleasure to have all these outstanding people speaking at the conference. These are really pioneers in cancer biology. We know that non-coding RNA are very important for development, for many aspects of development. So this conference is very interesting. For MDPI, for IGMS, for the journals in the company, it's a really good conference. We are partnering and we have relations with very important scientists. So it's really good. So we are very happy that people of this standing came for our first conference on this topic, and we hope this conference will develop further in the future.